from Faculty of Dentistry, University of Khartoum. I do have MSc of Biochemistry, Faculty of Medicine, University of Khartoum. Today, inshallah, we will study gluconeogenesis. Okay, at the end of this lecture, define gluconeogenesis. Gluconeogenesis. And you need to explain the process of gluconeogenesis or the steps of gluconeogenesis. Okay, let's talk about the gluconeogenesis. What is gluconeogenesis? Synthesis of glucose. So, gluconeogenesis, have a look to this synthesis of glucose pathway that will to synthesis. So, at the end of this pathway, we will have a glucose as it's a de novo that inside our body. Synthesis. Gluconeogenesis is the synthesis of glucose from non-carbohydrate substrates. Okay, what are the non-carbohydrates that can enter the glucose and end up with glucose? These substrates are amino acids, glycerol, lactate, propionate. Okay, glycerol, lactate, and propionate. Fine. What is the importance of gluconeogenesis? Generally, we do have some tissues, erythrocytes actually, which are completely dependent on constant glucose. C will utilize glucose. Also, the brain can utilize other substances than glucose. The main tissues that will completely or and constant on glucose is the erythrocyte. Okay, when the dietary carbohydrates are not sufficient, the blood glucose level is maintained by glycogenolysis in the liver, which is the process of degradation of the glycogen, which occur generally in the liver and in the muscle. But the glycogenolysis in the liver is the main pathway that can control the blood glucose, not as the muscle glycogenolysis, right? Which in general will utilize its own glycogen for itself, right? The muscle in generally will use its glycogen for itself, whereas the liver will utilize its glycogen, but it will use this glycogen to maintain the blood glucose level generally. Okay, so when we do have glucose level when the glucose level decrease, there will be activation of the glycogenolysis. Okay, also there will be activation of the gluconeogenesis during prolonged fasting. So first we will use glycogenolysis. If the fasting will sustain, this will increase gluconeogenesis reaction. Gluconeogenesis can occur in which sites? Gluconeogenesis primarily occur in the liver during fasting, okay? But also it can occur in the tubular cells of the kidneys, okay? The irreversible reactions in glycolysis are catalyzed by different enzymes that are unique to gluconeogenesis. You know that glycolysis contain 10 steps, right? But it contain some of these 10 steps are irreversible reactions because the enzymes that act in these steps will let the process or the pathway to go in a unique direction. There is other different enzymes in the gluconeogenesis okay, can let the reactions to be in the reverse way. Okay, generally the irreversible reactions in the glycolysis are characterized by hexokinase, of course, phosphorylcyclinase, and pyruvate kinase, as you already know, right?